So good morning. I'm going to talk to you today about a new insect pest. I'm guessing most of you, or almost all of you, haven't had a chance to see it yet in any of your fields, which is probably a good thing. But I think it's probably something that should be on your radar screens because it seems like it could be spreading, or at least our understanding of where it occurs is expanding every year. So let's dive into it here with the soybean gall midge. It's this tiny little fly. If you were to see one in one of your soybean fields, or if one landed on your arm or something, you probably wouldn't recognize it as anything other than, some other little gnat or something like that. But if you're kind of a bug geek, if you were to look close, you would see this alternating dark and light color pattern on the legs. That's pretty distinguishing. The wings have kind of this smoky mottled coloration. If you do have an infestation, this is what you're likely to see, the larvae. The larvae live inside the stems. They're maggots, right? So the larvae of flies are considered maggots. You can't tell the head end from the butt end. They don't have any legs. What's really distinctive about this insect is this bright orange coloration when they get more mature. In the early stage, they're transparent or white, but as they grow and get bigger, they take on that darker coloration. You can see a pretty good infestation inside of this stem here that was peeled open. So let's just see a quick snapshot of where we knew this thing to occur as of the end of this last summer. It first was detected in 2018 and 2019, indicated by the red and orange coloration here. Nebraska's been kind of the epicenter for the heavy infestations. The farmers there are really struggling with this pest. Fortunately, in Minnesota, the infestations haven't been that bad yet. You can see, up to 2022 in Minnesota, we didn't add any counties. 2021, we added quite a few. A lot of these were lower level infestations in green. What I want to point out is last summer, at the end of the year, Jan Knodel and her team documented what we're calling a tentative or unconfirmed, potentially, situation of soybean gall midge in Sargent County. I think there's a little discussion going on about, is it really soybean gall midge? Is it tentative? It looks like soybean gall midge. The damage it was causing to the plants was soybean gall midge, but some folks are saying since they weren't able to get the DNA and prove based on the DNA that it was soybean gall midge, we can't call it soybean gall midge. So I think Jan and her group are going to be doing some intensive sampling to hopefully get some larvae where they can get those DNA, but to me, I'm really thinking it is soybean gall midge because it's not that far from where we have known detections. So let's think about the life cycle of this insect a little bit. At this time of the year in the winter, it's a third instar larva, so one of those mature larvae that are bright orange inside of a cocoon in the soil. So they're going to be in the soil of last year's soybean fields in those cocoons. When spring comes around, those larvae are going to develop into the pupa and then the adult, and those adults are going to crawl out of the soil, and they're going to fly to the nearest soybean field. So here's last year's soybean. It's going to be corn this year. Here's this year's soybean. The adults are going to come out of the soil, and they seem kind of lazy. They're only flying, it seems, as far as they need to get to the nearest soybean. So that results in a pretty strong edge effect. So if you're scouting for this pest, the best chances of finding it are going to be in soybean fields, adjacent to last year's soybean fields, and then on the edges of those fields, right? Because they're just coming out, finding the nearest soybean, settling there. As the season progresses, they move farther into the fields. But once they come into those fields, they're looking for little cracks on the stems of the soybean plants. This insect doesn't have a way to inject its eggs into the plants. It needs to take advantage of other openings on those stems. So it'll lay its eggs into those cracks or near the cracks. When the eggs hatch, the larva or maggot will crawl into the stem and it'll start feeding inside. So here's a cartoon representation of some larvae inside the stem. And then once those larvae mature, they're going to drop out of the plant, go down into the soil, form that cocoon and pupa, and then again eventually the adult, and start that life cycle over again. In southern Minnesota, we're thinking we probably have 2, maybe 3 generations per year, and that'll probably change as we go farther north. You know, there'd be probably fewer generations the farther north we get. So I showed you some kind of cartoon representations of the injury. Here's a real picture. So those cracks that occur in the stems are usually low, near the soil surface. And once those larvae get inside there, they cause this darkened discoloration of the stem. It kind of distorts the shape of that stem a little bit. And if you were to peel back the outer layers of the stem, you could see the larvae inside. And just one thing to point out, you know, mentioning the V2, V3 growth stages, that's when we start to see infestation. That's not the only time that infestation occurs. That's just when it starts, and that's when we're thinking those cracks start to develop. We can see this insect infesting plants all the way through the vegetative and into the reproductive growth stages as well. So when they're feeding in the stems, they're destroying those tissues that are conducting water and nutrients within the plants, causing the plants to wilt and eventually die. You can see that in this picture here on the edge of a field down in southwest Minnesota. And they weaken the stems, causing them to break off or lodge. My colleague in Nebraska, who's been dealing with really heavy infestations of the soybean gall midge, has documented 100% yield losses on the edges of fields and up to 17 to 31% yield losses in the interior. So if you've got heavy infestations, this can be a very significant pest. Again, in Minnesota, the vast majority of fields that we're finding are not near economic levels of infestation. We can find it, but it's not that heavy of an infestation yet to be causing losses. Doesn't mean it won't happen. That's why we're keeping a close eye on it. We want to hear from farmers, consultants about where they're seeing it so we can get a better feel for what's going on with this new pest. So what does this insect attack? Its name is the soybean gall midge, so obviously it feeds on soybean. But in addition to that, it's been documented feeding on alfalfa and sweet clover in Minnesota and Nebraska. More recently, my colleague at the Lamberton Research and Outreach Center in southwest Minnesota did an experiment where he was placing potted plants of different kinds of legumes, different kinds of beans out into infested soybean fields, brought them back into his greenhouse, let them grow for a little bit, and he was able to find infestation on lima bean and some of the dry beans. And then we also found an infestation in a navy bean field. So just as of last year, we've expanded the host list, beyond these 3 species to now include 2 additional species of plants. So that's kind of the broad overview of the biology of this insect. Let's think about what options or what things might be going on in the environment that could help reduce the populations of this pest. One thing we think about for control of insect pests is biological control. So this is when we try to rely on some of the natural, predators like lady beetles or other kinds of predatory insects, parasitic insects, to knock the pest population down. There was no knowledge about this previously because the insect was first discovered in 2018, 2019. A lot of work going on with insecticides and other tactics. They're not looking very promising, so that's why we started looking into biological control. So we did this research down in southwest Minnesota where we do know of a heavy infestation. My students set up 2 fields, multiple locations in each field, some on the edge of the field, some on the interior of the field, sampled those fields throughout the growing season, and then we've got 2 years of data, 2021, 2022, but we're still kind of compiling the 2022 results, so I'll only show you what we have for 2021. The first thing I want to talk about is parasitic wasps. So when I say wasp, I'm not talking about yellow jackets and things like that that are going to fly around and sting people. These are tiny, tiny little insects, and their stingers are modified to be an egg-laying tool. So they use their stinger to inject their eggs into whatever insect they're attacking. So in this case, the soybean gall midge. So we went out into fields on this farm, collected infested soybean stems, put them into these emergence cages in our lab, and then we checked those cages every day to see if any soybean gall midge adults would come out and to see if any of these parasitic wasps would come out. And we got super excited when we started finding one of these wasps. So that's an extreme close-up here. The interesting thing is, this wasp, its DNA doesn't match any other known wasp. And if you look at the morphology of it, different details on its body shape and things like that, it doesn't match any other known wasp. So not only do we have a brand new species of pest, the soybean gall midge, in Midwest soybean fields, we've got a new species of wasp. And for me, as a soybean entomologist in the Midwest, this is pretty exciting, right? Because it's not too often that you get a chance to discover a new insect. That's usually for the folks that are working in the tropical rainforests and stuff like that. So in addition to trying to rear insects out of infested plants, we've been doing some work where we've dissected the larvae out of the plants, preserved them, and then we're looking at those larvae, doing tests to see if they have the DNA of the wasp inside of them. So if you got a soybean gall midge larva and it has wasp DNA inside of it, that means that it's been attacked by one of those wasps, right? Either as the egg of the wasp or the larva inside of it. So here's a picture to give you a bit of a size reference. So that's the soybean gall midge adult, the larva, and I think at your lunch session you're going to have like a hands-on workshop, and Anthony Hansen will have some displays that I put together where you can see how small the soybean gall midge is, and then just remember that this wasp is considerably smaller than that. So on this graph, we've got time through the growing season in 21 across the bottom. This is proportion parasitism, so how many of those soybean gall midge larvae were attacked by the wasp. The blue bars are the results from the molecular test. The pink bars are from those emergence cages where we were rearing the insects out. Not going to get bogged down in the details here, but we've got more parasitism early in the season and then later in the season. So that suggests there might be a couple different generations of the wasp. And this molecular tool seemed more sensitive than the rearing. We were able to detect higher levels of parasitism. So this is just 1 year of data. We're going to continue this, and after I talk about the predator work, then I'll talk about kind of why are we interested in these in biological control. So for the predators, a lot of times when we think about predators, we might think about spiders or lady beetles that could be on our soybean plants eating our aphids. But there are a lot of different predators that scurry around on the soil surface in our fields. So to catch those, we use these traps where we bury cups in the soil, put some antifreeze in there, cover it with a little foam plate to keep debris or rain from getting in. And then after leaving those out for 24 hours, we were finding lots and lots of insects in there. And it was pretty much just one kind of insect. This one beetle here, we don't need to worry about its scientific name, but it's a kind of ground beetle. And they're very well-known predators that are known to attack and play a role in managing pests in other systems. So this was really promising to see that we've got a well-known predator associated with this new pest, but it left us wondering, well, if it's there, is it feeding on it? So we set up a pretty simple experiment in the lab where we got petri dishes, we put in some of the soybean gall midge larvae, we put in some beetles, and then checked the dishes over time to see if the predator would eat this prey. And sure enough, they did. Here's one of the beetles bunching on a soybean gall midge larva. Here's some of the numbers, showing that they will indeed feed on soybean gall midge. But we know soybean gall midge isn't the only thing out in our soybean fields that it could eat. What if we had a soybean aphid infestation? Then that predator is going to have a choice. Well, do I eat the soybean gall midges or do I eat the soybean aphid? So we started wondering, would they have any preferences? So my student set up another petri dish study where she put in different numbers of soybean gall midges or soybean aphids, left them for a certain amount of time, came back and checked them to see how many of each kind of insect they ate. And this gets a little complicated, so I'm just going to try to make it as simple as possible here. On this graph, this is the ratio of the prey offered, the aphids, the soybean gall midges and the aphids, and this is the ratio that were consumed. But kind of the easiest way to think about it is if the data fall out in this green area, it means that predator has a preference for the aphids. If it falls out in the kind of orange colored area, it means it has a preference for the soybean gall midge. And kind of surprisingly what we saw, this predator did indeed show a pretty strong preference for the soybean gall midge compared to the aphids. So that's kind of cool, but it's super artificial, right? We've got these predators locked up in small petri dishes, and they can either eat this insect or they can starve to death and die. So, I mean, sure enough, they were eating it, right? So to get to some more real world information, all those predators that we were collecting out of those cups in the field, we were saving those, and then we're gonna do some molecular tests where my students are doing this right now, they're dissecting out the guts, like the stomach, digestive system from these beetles, and then we're doing those genetic tests to see if these beetle guts have soybean gall midge DNA. And then that'll tell us if these predators actually are eating soybean gall midge in the field. Preliminary data are showing that they are, but we just don't have enough of the data processed yet to say like what rates of predation are happening or how much control could we maybe expect from these predators. So shifting gears here away from the beneficial insects. Again, we live in northern states. We know our winters are pretty cold. One good thing about that is that these cold temperatures can kill off certain kinds of insect pests. That's why we don't have it as bad in our soybeans as some of the guys in the southern states, right? It's a nightmare down there for managing insect pests in soybeans. So one thing we were wondering is how well can the soybean gall midge survive our cold winter temperatures? And this has implications for how far north might this pest spread, or predicting infestations from one year to the next based on how cold it gets over the winter. So what we did is we went into some soybean fields toward the end of the year, collected infested stems, dissected larvae out of those stems again, and then through some trial and error, we were able to figure out how to trick these larvae into producing their overwintering stage, which again is that third instar larva inside of the cocoon. So then once we had those cocoons, we took them and put them into what I'm calling kind of simulated fall or winter conditions, what they would be experiencing before winter really sets in, to let them kind of acclimate to winter. So 37 Fahrenheit, 55 Fahrenheit, short day lengths. Would have been nice to get some colder temperatures, but that's all we could do with the equipment we had. And then we held them at those conditions for 1 month or 2 months. And then after those time periods of 1 month or 2 months, we did 2 different kinds of tests to see what kinds of temperatures these insects could handle. One of them we call lethal temperature. So we take the insects, we cool them down to different targeted cold temperatures. And then once they hit that, we pull them out, warm them up, and see how many are dead, how many are alive. And then the other test we can do is to see at what temperatures are these insects freezing. And for insects, a lot of them produce some antifreeze compounds in their body, which allow them to freeze below the freezing point of water. So we call it super freezing, or super cooling, sorry. And we can hook the insects up to these little probes that are connected to a computer, and then we can monitor their temperatures as we cool them down. So as we're cooling down an insect, eventually we'll see a little spike in heat, and that indicates when they're freezing. When something freezes, it releases a little bit of heat. So that lowest point before that, that's the freezing point. So here's some data on the lethal temperature. So what's their survival when they're exposed to these different cold temperatures? This graph on the left is for individuals that were acclimated or held at 37 Fahrenheit, this for 55 Fahrenheit. These are the temperatures that we cooled them down to on the bottom. And for both of these conditions, we see that their proportion survival was really good from 14, 5, and -4. But once you got below -4, it dropped off really fast. And coincidentally, that steep drop-off matched up really well with where these insects were freezing. So right around -8, -9 Fahrenheit. Well, what does that mean in the real world, right? We know our air temperatures can get below that, but what about in the soil where these things are spending the winter? We know the snow can provide some insulation. We know the soil can provide insulation. So I dug up just a little bit of soil temperature data. We're trying to get some more. This is data from 2 inches deep in the soil, and for each month on the graph, we plotted the lowest temperature that was experienced in that month. The different colored lines are the data from, I think, 6 different years, and then that thicker black line is a 20-year average. Unfortunately, that kind of critical temperature where this insect is freezing and dying is quite a bit lower than what we were experiencing in the field, at least in southwest Minnesota. Okay. We're still trying to get some data from farther north in Minnesota, but as far north as we go, I'm guessing we're probably not going to be crossing this line. So to me, that suggests we're probably not going to be able to count on our cold winters to knock back these populations. But kind of the one catch here, remember, is that this was just a very short-term exposure to the cold. We were cooling them down, and then as soon as they hit that targeted temperature, we remove them to warm them up. So what we want to try to do next year is cool them down to these temperatures and then hold them there for some extended period of time. Because we know for other insects, they might be able to survive a set temperature if they're there just for a little bit, but if you hold them there for a long enough time, they'll eventually die. And in the field, that's what we experience, right? Our temperatures don't just drop and bounce back up, right? They drop and they stay down there for a while. So here's a pretty heavily infested stem. Just kind of wrapping things up. So soybean gall midge, it's a new threat. I should have added North Dakota to the list here too. For Minnesota, North Dakota, really the Midwest, right? We've seen that known range expanding on that map that I showed. Again, if you're scouting for this pest, the edges of fields that are near last year's soybean are the highest risk. If you're scouting, you'd want to be looking for those lesions at the base of the plant, right? Kind of open up the canopy, look down to the base of the plant, look for those darkened lesions, wilting plants, dying plants, lodging. If you're seeing that, pull up some of those plants, start peeling open the stem, and look for those orange larvae. If they're there, they're really obvious, right? But there are some other diseases and other things that can cause that discoloration at the base of the stem. So I didn't have time to go into the details, but for chemical control, insecticides, right? When we have a new pest, a lot of times that's what we're thinking about or going to for management. And my colleagues across Minnesota, South Dakota, Iowa, and Nebraska, they've all been doing insecticide efficacy trials for this pest, and none of them are showing very strong results yet. Seed treatments, foliar applications, it just doesn't appear that we're going to be able to use insecticides as a silver bullet for controlling this pest. So that's why we're looking at some of these other things like biological control, right? To get more toward integrated pest management, right? If we can maybe use insecticides to knock them back a bit, but try to do what we can to promote some of these predatory insects or parasitic wasps, maybe together we can keep the populations at a tolerable level. And again, they appear very cold hardy, so it's probably not very likely that our winters are going to slow them down much. And then just some acknowledgments here. Definitely want to express my gratitude for the soybean grower associations at the state level, regional level, for all the support for this work. If I have time for questions, I'm happy to try to address any. Yep, in the back.
Have you noticed the difference between, say, fall tillage and no-till, even though it's kind of early in this breakout?
Yep, so the question was about tillage, fall tillage, spring tillage. We know with some pests, tillage can do a good job in knocking back the populations. For this insect, my colleague in Iowa, Aaron Hodson, for 2 years, has tried an experiment to look at tillage, looking at fall tillage, looking at spring tillage, different kinds of tillage, and sure enough, the insect wouldn't cooperate. One of the best ways to get rid of an insect pest is to have a university person come out and try to set up an experiment, and sure enough, that dang pest won't show up. So she's gotten skunked 2 years in a row where there just weren't enough insects in her plots to really get a feel for things. Looking at some of the other gall midges, I think it's kind of hit and miss. Sometimes tillage, there are other species of gall midges that are pests in other crops, and sometimes tillage will help with some of those. Sometimes it won't. Sometimes it depends on the timing and the kind of tillage. So I guess all I can really say is stay tuned. Hopefully Aaron will have some better luck. Yeah.
We still
Kind of-
have time for a few more questions.
So one interesting thing, kind of related to tillage, my colleague in Nebraska, Justin McMecken, has been working with hilling. And what it is basically, you know, planting your beans and then going through with an implement and piling up soil at the base of those plants to cover up that, you know, that bottom 2 to 3 inches of the stem where those cracks are occurring. And he's getting really good results with that. It does protect those plants, but how the heck would we ever, you know, implement something like that in a, you know, any kind of efficient way, right? There's some work with screening soybeans to see if any varieties have resistance to it. That's being led up by the soybean breeder in Nebraska, collaborating with folks in other states, screening hundreds and hundreds of different varieties. And it sounds like they do have a couple varieties that are showing some resistance. So that's good. But we all know that these breeding programs are really slow. It just takes a long time to identify whatever traits we're looking for, and then to cross those into, you know, high-yielding, well-adapted varieties. So that's something promising, but it's not going to be available anytime soon. Yeah. So as you can see, we're struggling with this pest, right? We just don't have too many solid tools for you. So if any of you have or hear about heavy infestations, let us know, because the more research sites we can get out, the more information we can hopefully produce to get back to all of y'all. Was there another question?
If you expanded your location map to a larger area, get beyond Nebraska, is it unique only to this area, or are there other hotspots?
Yeah, so the question is, you remember that map I showed that was kind of focused on the Midwest, centered around Nebraska? If we zoomed out and looked over the US or the whole world, would we see it anywhere else? And the answer is no. This insect has not been detected anywhere else in the world. You look back at all the scientific literature, there's no mention of it from anywhere else. So I guess we're just that lucky in the Midwest to have this thing. We're not even certain if it's an exotic species that could have come over from somewhere where it, you know, maybe it was from somewhere else in the world, never been detected there and came here, or if it's always been here, maybe feeding on some native legumes. No one ever noticed them until they started deciding to feed on soybean. Yeah.
That wasp, was that unique? You were the first one to find it in this area, or unique?
Ever. Yep, so not only is the pest insect brand new, no one's ever seen it anywhere in the world, this wasp is the same. Yep. Not sure how much, okay.
Is transmission only mobile from the insect, or can it be transmitted via tillage and other methods?
Yeah, so the question is about, you know, how mobile is this insect, or could tillage move it around, or, you know, transporting equipment or something maybe. The insect itself, you know, they do fly, but I think it's generally thought that gall midges, this kind of group of insects, are not real strong flyers, but they're small, so they can get up, you know, and be blown around in the winds, or if they get up high enough, like in the jet streams, like think of soybean aphids, right? We know those can be moved across states. As for kind of human-assisted movement, I guess it's possible. No one's done any research yet, but, you know, there are other soil pests, like some of the nematodes with cysts and things that can get moved around. These cocoons are a little bigger than that, so it might be less likely. Honestly, I'm not too sure. I mean, if you were moving around large volumes of soil, I think it certainly could, you know, but if it's just smaller amounts on equipment and things, I think it's much less likely, but I can't say it's not a risk, right? Yeah. Okay. Well, thank you,
Any more
everyone.
questions? Perfect, thank you so much.
Yep.